In this session, our guest speaker will be discussing his presentation titled M2GEN's ORIEN Avatar Program Accelerates the Discovery, Development, and Delivery of Comprehensive Oncology Research with a Custom xGen Exome Research Panel. Dr. Hampton is the VP of Bioinformatics and Biostatistics at M2GEN and is responsible for leading and building out the bioinformatics and biostatistics teams to support the Avatar project as well as collaborative projects between M2GEN, Pharma, and ORIEN. Prior to joining M2GEN, he led the platform informatics team at the Center for Molecular Oncology at Memorial Sloan Kettering Cancer Center. I'd like to thank Dr. Hampton for his time in putting together today's presentation. We will now begin today's session.
Hello, my name is Oliver Hampton, and I am the Vice President of Bioinformatics and Biostatistics at M2GEN. Today I will be presenting. The title of the presentation is M2GEN, ORIEN's Avatar Program Accelerates the Discovery, Development, and Delivery of Comprehensive Oncology Research with a Custom xGen Exome Research Panel. I'd like to start today with a brief introduction about M2GEN, who we are, and what we do. M2GEN is an oncology informatics company founded in 2016 to operationalize the Total Cancer Care Protocol. The Total Cancer Care Protocol, or TCC, is one of the world's largest observational studies in cancer and lies at the heart of everything we do here at M2GEN. The TCC enables us to do two fundamental things that are crucial to our operations. One, it provides us with a standardized mechanism for consenting patients in a centralized, harmonious fashion. And two, it enables us to abstract, collect, and aggregate both clinical and molecular data in a centralized way, thus enabling data capture to be standardized. The TCC protocol is implemented at 18 of our nation's leading cancer centers, where patients consent to share their clinical and molecular data throughout their cancer journey. Patients' data is aggregated and shared across these 18 academic cancer centers, which is called the Oncology Research Information Exchange Network, or ORIEN, enabling cancer research collaboration and intermember projects between those institutions. Our core offering as a company is the ORIEN Avatar dataset, something I will explain greater in detail. M2GEN sits at the center of this patient-driven ecosystem, wherein the Total Cancer Care Protocol is implemented across the ORIEN network, allowing M2GEN to create a unique, industry-leading data set while facilitating data-sharing partnerships and fostering collaboration between ORIEN members, all in a concentrated effort to accelerate discovery of new therapies for cancer patients. This is a patient-driven learning health system. M2GEN bridges the gap and acts as a conduit between patients and clinicians, academia, and our pharma partners. The ecosystem is a patient partnership. Patients consent to the TCC protocol, agree to be followed throughout their lifetime, enabling collection of longitudinal clinical and molecular data. These patients donate specimens to be used to generate that molecular data. They also consent to be recontacted for future studies. This allows us to perform prospective data collection and obtain retrospective data from the patient's chart and medical history, constantly enriching the data set, both through the addition of new patients and by continually adding and updating the records of our existing patients. M2GEN follows these patients throughout their entire cancer journey. Importantly, we continue to add to the growing roster of these total cancer care patients at a clip of 25,000 to 30,000 patients a year. The Total Cancer Care Protocol is implemented at 18 of the nation's leading cancer centers across the continental United States. This gives us enormous patient diversity. For example, there's a high vet population at Martha Cancer Center and George Washington, high African American population at Emory and Morehouse, and high Hispanic American populations at UNM and USC Norris. We endeavor to grow the avatar by at least 1 to 3 sites per year. The next section of the presentation will describe the core offering by M2GEN. This is called the avatar. It is a curated data solution. In consultation with our partners, M2GEN has identified cohorts of cancer patients at high risk for disease progression and those who have limited treatment options. These are what we dub avatars. These are the patients who will benefit the most from the insights derived from the avatar program. Patient cohorts span both solid tumor and heme malignancy. M2GEN collects longitudinal clinical data for all of our patients in avatar. The avatar data model includes over 600 data elements, including demographics, diagnosis and staging, and medical treatment data that allows retrospective analysis for lines of treatment and outcomes data. Clinical data is refreshed at least every 6 months or sooner for some indications. In addition to the clinical data, M2GEN performs deep sequencing on all of our avatar patients. Each patient has tumor and germline whole exome sequencing as well as RNA sequencing. Currently, there are over 18,000 patients in the avatar program. Avatar is an ever-expanding clinical genomics data set, which grows at a rate of between 5,000 and 10,000 patients per year. There have been 2 major transitions since inception of the avatar program. They're depicted here with the dotted lines. The first was a sequencer shift from Illumina HiSeq to Illumina NovaSeq. That happened earlier than the transition of our whole exome capture probe set. The original part of the avatar program utilized a Roche or M2GEN SeqCap Easy Exome version 3 probe set, but we've since made a transition to the IDT xGen Exome Research Panel version 1 with additional custom probe design as a spike-in. The transition from the Roche SeqCap Easy Exome to the xGen Exome Research Panel with custom spike-in resulted in a few changes, mainly a 34% reduction in targeted regions, an introduction of double-covered areas across approximately 440 cancer genes, and custom spike-in probes for intronic regions for structural variation detection, and cancer-related promoter and enhancer regions. The transition from the Roche to IDT capture probe sets resulted in a number of specification capture metrics that we will review over the next few slides. Looking at the tumor whole exome sequencing data across the entire ORIEN avatar dataset, we see 3 distinct populations. The first, on the far left in gray, is the Roche Easy Exome sequenced on the Illumina HiSeq sequencer. In the middle in orange, again is the Roche or M2GEN SeqCap Easy Exome, but sequenced on NovaSeq. And lastly in blue is the IDT xGen Exome Research Panel, also sequenced on the Illumina NovaSeq platform. It's important to note that the read counts per tumor capture increased from HiSeq to NovaSeq. So further evaluation of sequencing metrics will only be evaluating the differences in the probe set and stabilizing across the NovaSeq platform. No additional analysis on the HiSeq will be made. Looking at the mean target coverage and capture uniformity metrics between the Roche and IDT custom spike-in, we see 2 distinct things. 1, we see a higher mean target coverage with IDT due to the reduction in targeted region compared to Roche. Second, we see better capture uniformity with IDT, resulting in greater than 99% of all tumors sequenced having an 80% of target bases covered at 100x or greater. Lastly, M2GEN's ORIEN avatar design consists of 37.6 megabases of single and 1.3 megabases of double-covered capture probes. The custom-designed double-covered regions perform exceptionally well with a 2.2-fold coverage rate compared to the single-covered regions. For the last part of the presentation, I would like to highlight a couple of use cases that look into the somatic mutation detection sensitivity of M2GEN's custom IDT probe design. The development of liver cancer, specifically hepatocellular carcinoma, or HCC, is promoted by the accumulation of specific driver somatic mutations. These are depicted in this slide's oncoplot. These driver somatic mutations arise in CTNNB1, TP53, and the TERT promoter region, also referred to as the 5' flank region. The TERT promoter driver mutations were not identified when sequenced with the Roche CCAP-EZ exome. However, when we switched to the IDT xGen Exome Research Panel, these were indeed covered and have resulted in accumulation and detection of these specific onco-related promoter region mutations. Sequencing coverage directly impacts somatic mutation calling sensitivity and detection rates. For example, 24% of EGFR T790M somatic mutations from the Park et al. Paper are under 5% VAF. So the higher the depth of coverage, the higher the sensitivity to detect lower and lower VAF mutations. Evaluating the detection rates from these 12 low VAF somatic hotspots in the ORIEN avatar dataset between Roche, NimbleGen, and IDT, we see statistically significant higher detection rates using IDT with the double-covered custom spike-in probes that do indeed cover all of these 12 somatic mutational hotspots. Looking at one of these hotspots, specifically BRAF V600E, we can evaluate the differences seen between the Roche, NimbleGen, and IDT capture probe designs. What we see on the histograms on the left panels are the variant allele fraction distributions as well as the alt, or the reads containing the specific mutations, allele depth on the bottom histograms. What we see in the IDT double covered regions versus the Roche, NimbleGen single covered regions is that we detect a far larger proportion of low VAF somatic mutations than we do in Roche, and this is being driven by a higher alt allele depth at those locations. On the right, we see a violin plot of those VAF mutations distributed between those two separate probe sets. In conclusion, M2GEN's ORIEN Avatar program has benefited from the transition to the IDT xGen Exome Research Panel with custom spike-in probe sets, specifically enabling higher coverage in actionable oncology genes, also allowing us to specifically probe for intronic regions for structural variation detection, as well as cancer-related promoter and enhancer regions. We see improvement in target coverage and uniformity metrics also when transitioning from Roche to the IDT xGen Exome Research Panel version 1. Finally, the somatic mutation detection sensitivity improved across the double-covered oncology actionable genes, also using the IDT panel. Thank you for your time.